Hello, welcome to Biology, the scientific study of life. Supporting cells, glia. Glia from a Greek word meaning glue are supporting cells that are essential for the structural integrity of the nervous system and for the normal functioning of neurons. In the mammalian brain, glia outnumber neurons 10-fold to 50-fold. There are several types of glia in the brain and spinal cord, including astrocytes, radial glia, oligodendrocytes or oligodendrocytes and Schwann cells. As a group, these cells do much more than just hold neurons together. In the CNS, astrocytes provide structural support of neurons and regulate the extracellular concentrations of ions and neurotransmitters. Some astrocytes respond to activity in neighboring neurons by facilitating information transfer at those neurons. Synapses. Scientists hypothesize that this facilitation may be part of the cellular mechanism of learning and memory. Astrocytes adjacent to active neurons also cause nearby blood vessels to dilate, which increases blood flow to the area, enabling the neurons to obtain oxygen and glucose more quickly. During development, astrocytes induce the formation of tight junctions between cells that line the capillaries in the brain and spinal cord. The result is a blood-brain barrier, which restricts the passage of mass substances into the CNS, allowing the extracellular chemical environment of the CNS to be tightly controlled. In an embryo, radial glia from tracks along which newly formed neurons migrate from the neural tube, the structure that gives rise to the CNS. And both radial glia and astrocytes can also act as stem cells generating neurons and other glia. Researchers view these multipotent precursors as a potential way to replace neurons and glia that are lost to injury or disease. Oligodendrocytes in the CNS and Schwann cells in the PNS are glia that form the myelin sheaths around the axons of many vertebrate neurons. The structure of myelinated PNS axon is shown in this figure. Notice that. Neurons become myelinated during development when Schwann cells or oligodendrocytes grow around axons, wrapping them in many layers of membrane, somewhat like a jelly roll. These membranes are mostly lipid, which is a poor conductor of electrical currents. Thus, the myelin sheath provides electrical insulation of the axon, analogous to plastic insulation that covers many electrical wires. In the disease multiple sclerosis, myelin sheaths gradually deteriorate, resulting in a progressive loss of body function due to the disruption of nerve signal transmission. Ion pumps and ion channels maintain the resting potential of neurons. All cells have an electrical potential differences voltage across their plasma membrane. This voltage is called the membrane potential. In neurons, the membrane potential is typically between 60 and 80 millivolts when the cell is not transmitting signals. The minus sign indicates that the inside of the cell is negative relative to the outside. Explains how electrophysiologists measure a cell's membrane potential. Notice that the resulting potential, the membrane potential of a neuron that is not transmitting signals, is called a resting potential. In all neurons, the resting potential depends on the ionic gradients that exist across the plasma membrane. In mammals, for example, the extracellular fluid has a sodium ion in a positive concentration of 150, notice that, 150 millimolar, and a potassium ion, K positive concentration of 5 millimeter, 1 millimolar each liter, equal 10 to the power minus 3 each liter. In the cytosol, the Na positive concentration is 15 millimolar, and the K positive concentration is 150 millimolar. Thus, the Na positive concentration gradients expressed as the ratio of outside concentration inside concentration is 150, 15, or 10. The K positive concentration gradients 5 to 150 or 1 to 50, 30. There are also anion gradients, but we will ignore them for the moment. The Na positive and K positive gradients are maintained by the sodium-potassium pump. The fact that the gradients are responsible for the resting potential is shown by a simple experiment. If the pump is disabled by the addition of a specific poison, the gradients gradually disappear, and so does the resting potential. It is particularly challenging to understand how the resting potential arises from ionic gradients, but it is an essential step in learning how neurons, including our own work. We start with a model of mammalian neuron consisting of two chemical chambers separated by an artificial membrane. The membrane contains many ion channels that allow only K positive to diffuse across a membrane. The membrane to produce concentration gradients for K positive like that of mammalian neurons. We added 150 millimolar potassium chloride KCl positive to the inner chamber and 5 millimolar KCl to the outer chamber. Like any solute, K positive tends to diffuse down its concentration gradients from an area of higher concentration inner chamber to an area of lower concentration outer chamber, but because the channels are selective for K positive, outer or chloride ion Cl minus cannot cross the membrane. As a result, a separation of charge voltage develops across a membrane with an excess of negative charge on the side of the membrane. Facing the inner chamber, the developing membrane voltage opposes the efflux of K positive because the excess negative charge attract the positively charged K positive. Thus, an electrical gradient builds up whose direction is opposite that of the concentration gradients. When the electrical gradients exactly balances the concentration gradients, an equilibrium is established. An equilibrium, there is no net diffusion of K positive across the membrane. Notice that. Call it the equilibrium potential K, notice that, E ion. E ion, notice that, is given by a formula called the Nernst equation for an ion with a net charge of 1 positive, such as K positive, at 37 Celsius degree. The Nernst equation is like this. Notice that. The Nernst equation applies to any membrane that is permeable to a signal type of an ion. In our model, the membrane is permeable only to K positive, and the Nernst equation can be used to calculate EK. The equilibrium potential for K positive is like this. Notice that. Notice that the minus signs indicates that with this K positive concentration gradients, K positive is at equilibrium when the inside of the membrane is 92 millivolt voltage more negative than the outside. Now imagine that we change our model neuron by using a membrane that contains ion channels selected for Na positive. We also replace the contents of the chambers to produce a concentration gradients for Na positive like that of mammalian neuron. 15 millimolar sodium chloride NaCl in the inner chamber and 150 millimolar NaCl in the outer chamber. Under these conditions, the Nernst equation can be used to calculate the equilibrium potential for Na positive like this. Notice that. The plus sign indicates that with this Na positive concentration gradients, Na positive is at equilibrium when the inside of the membrane is 62 millivoltage more positive than the outside. How does a real mammalian neuron differ from these model neurons? The plasma membrane of a real neuron at rest has many potassium channels that are open, but it also has a relatively small number of open sodium channels. Consequently, their resting potential is around 60 to 80 millivolt voltage between K positive and Na because neither K positive nor Na is at equilibrium. There is a net flow of each ion occurring across the membrane at rest. The resting potential remains steady, which means that the K positive and Na positive currents are equally and opposite. The reasons the resting potential is closer to Ek than the Na. Notice that. Notice also is that the membrane is more permeable to K positive than to Na positive. If something causes the membrane's permeability to Na positive, to increase the membrane potential will move toward Na positive and away from Ek. This is the basis of nearly all electrical signals in the nervous system. The membrane potential can change from its resting value when the membrane's permeability to particular ions changes. Sodium and potassium ions play major roles, but there are also important roles for chloride Cl- and calcium Ca to the power 2 positive ions that follow the same rules. Gated ion channels. The resting potential results from the diffusion of K positive and Na positive through ion channels that are always open. These channels are said to be ungated. Neurons also have gated ion channels which open or close in response to one of three kinds of stimuli. Stretch gated ion channels are found in cells that sense stretch and open when the membrane is mechanically deformed. Ligand gated ion channels are found at synapses or synapses and open or close when a specific chemical such as a neurotransmitter binds to the channel. Voltage gated ion channels are found in axons and the dendrites and cells body of some neurons as well as in some other types of cells, and open or close when a membrane potential changes. Okay, thanks for listening.